Welcome to the Soil CRC's Building Technical Capacity for Improved Soil Management webinar series. Understanding the mechanisms of soil microbial function and their role in cropping systems. In this video, Southern Cross University PhD student Cameron Copeland shares the methodology and goals from his ongoing PhD project researching soil microbial function and its role in cropping systems, supported in part by the Soil CRC. Cameron discusses microbial function and diversity and the effects of environment and management for improved microbial function.
As the title says, I'm understanding the mechanisms of soil microbial function and their role in cropping systems. And I'm supervised by Terry Rose, Michael Rose, and Lucas Van Sweeten, and all three of those guys are involved with the Soil CRC. There are quite a few practices within cropping systems in Australia that influence soil biology or microbes in soils. The first is tillage. So conventional tillage, what happens is it's the preparation of the seed bed. It stops the competition of weeds with the microbes. And during this time, there's sort of like a burst of microbial activity. What's happened is the soil organic matter that some of those microbes might not have had access to, when the tines have gone in, they've turned up all that soil. The microbes, they just have all this access to all this carbon storage that wasn't in their vicinity at the time, and they just go gangbusters. So during conventional tillage, there's an immediate burst of microbial activity. However, this subsides fairly quickly, and what may have happened is that that tillage might have broken up sort of microbial networks that were pre-existing and working harmoniously and at equilibrium, as we're talking about earlier. And then, so there's conservation tillage, which is as long as about 30% of the soil biomass is left on top of the then that's what's considered conservation tillage. And then there's no tillage, and no-till practices, while are fantastic for microbes because they get to stay in that state and keep working and undisturbed. They also, the carbon pool can become stagnant, and less carbon can be sort of become available, so they can start eating into that carbon pool. Your crop management, so your crop rotation, where you cover cropping, intercropping, and companion cropping. Obviously, monocropping would have a negative effect because it's the same carbon sources going in over the same time. And what that does is that reduces your microbial diversity down to only a small set of microbes that feed off what's being fed by that same plant over and over and over again. And this has had to sort of pathogenic microbes come into play. And at the same time, it's allows sort of diseases to come through, and then precision sowing. So they're starting to realize that if you sort of sow your crops too close together, that same plant is drawing from a common microbial pool, and then the plants start competing with each other for those microbes. And sometimes there's not enough nutrients in the soil to allow the sort of expanding microbial microbiome to service both plants. So they're trying to tap from the same sort of nutrient source. So I know CSIRO and I know DPI are both looking at not only row spacings, but how far apart the plants need to be. And it's obviously different for different crop species. And that's a huge leap forward in understanding that if crops are sown too close together, then those microbes are, feeding off the same plant. And then obviously our chemicals and our inoculants, so herbicides, pesticides, particularly fungicides, can be very detrimental. Now, fungicides can be targeted. However, they can wipe out a large portion of fungi because fungicides aren't necessarily species specific. They tend to be sort of more at the higher levels of a target. So it might be like a family, so you get family, genus, species. So once it hits that one family, that's a whole bunch of different fungi that have been knocked out. Then there's obviously your inoculants, so your microbial sprays. And as we're doing this project, we're trying to figure out whether they actually reach their intended target. But another way to do this is seed coating. So seeds are coated with a microbial inoculant, and then they're there from the start, from germination. Yeah,
Cameron,
sorry.
Can I just ask a question about if you go back to the other slide with the chemicals and inoculants?
Yep.
We're getting a bit of a shift towards because of the concerns around microbial activity in the soil and understanding their function in cycling nutrients and making some nutrients available. We are getting a bit of a shift towards some landholders not wanting to use fungicides like seed dressings in particular. So, and then also this, so that's sort of one question, I suppose, if you'd like to consider, and also these microbial sprays and seed coating. So I've got landholders who are, mixing up their own inoculants or extracting, worm extract out of composts and using that as what they're calling as an inoculant or a seed coating. So we're getting this language being used for different, traditionally seed coatings were fungicides or a rhizobium inoculant, and now people are mixing up their own, what they're calling seed coatings and seed dressings, which they're believing is adding biology and
to the seed,
to the soil for a benefit of nutrient cycling or boosting the microbiology in the soil. I mean, do you have a comment about that?
Yes, that's I guess seed coating for me isn't what's being used, it's the process. So you can coat your seeds in anything you like, and to say that your seed coating doesn't have any connotations, both positive or negative. It's simply you are coating your seeds with something. Now, I know that it's particularly in the wheat industry, it's very common to coat your seeds in something like gaucho, just to protect the seed for those early germination times. And that does have an effect on the microbiome immediately around the seed. But you're right, if you were to coat it with a sort of microbial community, and you're talking about they're not using fungicides anymore, or they're coming up with their homemade microbial inoculants. As you're about to find out in a couple of slides later, unless they specifically know the diversity profile that they got to in their soils to begin with, you don't know that the microbes that you're adding into that soil benefit that soil type and have the right conditions for those microbes that you're adding. So you might add those microbes in, and then a week later they're all gone. They didn't survive. It's the wrong type of conditions for the microbes that you've added. They might go gangbusters. You don't know until you've taken a sort of a sample and you've tested that microbial diversity to start with, and you're sure that the microbes that you're adding are sort of able to withstand the conditions or the crop management practices that you have, as well as the soil type. Then it may last that one season, and then if you don't put a cover crop on, that particular microbe that you've added might not survive because it needs a constant feed of nitrogen or carbon, or it needs it might be an ANF fungi, so it needs a root system. If it doesn't have a root system to hang on to, it's gone within the next growing season. And then you've sort of run into this same problem with farmers of having to put chemicals on chemicals. If you don't look after your management practices, you might have to keep adding your microbes into your soil. And as we're starting to find out at the moment, that microbial inoculants aren't cheap. And I think it's on average about $40 a litre. And so you've still got a cost there to add to your crop. I think, so it's the Australian Inoculant Research Group at EMAI. They've done a approximate calculation that if we were to stop using nitrogen fertilizer and switch to complete biological fixation of N in our soils, it would save Australian growers to the tune of $4 billion. So it's great in theory to make that switch, but whether that happens in real time, that's easier said than done. It's the analogy I use is like trying to convince Australia as a country to switch to solar energy, and we're dealing with something much more complex than a solar panel. We're dealing with a live, forever changing dynamic system of microbes. So to convince that the whole of growers to no longer put, sort of fertilizer into their soil and make it completely microbial, that's a huge task. And at the same time, we're still not 100% sure that it's going to work based on because each growing site is different, each management practice is different, cropping types are different. So there's almost has to be a separate microbial plan per grower, per site. So even a field that's about 1 kilometre away would have a completely different microbiome, different soil types, different, and that's going to affect what microbes you should be putting in your soil. And so the soil microbes are involved these are the sort of key soil functions. So you've got the biochemical regulation of plant hormones and diseases. You've got your soil organic matter, turning over of the organic matter and carbon storage. Nutrient cycling is probably the biggest one. Soil structure. So they're involved in sort of aggregation, and then soil water availability. So I was just going to go a little bit into not exact mechanisms of how they do this, but just a brief overview. So if I lose you here at any point, just stop me and I could sort of further explain it. So not all microbes are beneficial. And if you have a sudden or increase in microbes in your microbiome, in your soil, the chances are that there's going to be pathogenic bacteria and fungi. Same with the monocropping. So if you sort of crop over and over and over and over again, eventually there'll be pathogenic fungi and bacteria that go, hang on a minute, I really like this food source, and they start, out competing. But there are obviously beneficial ones. So you've got your plant growth promoting rhizobacteria, and as you can see here, they come in sort of the Pseudomonas, Bacillus, and Serratia varieties, and they prevent pathogenic bacteria through nutrient competition. So PGPR are very good at outcompeting pathogenic bacteria. They tend to be more efficient in taking in soil nutrients. Then you got your AMF. Now they tend to attach themselves to roots, and they stimulate a phytohormone response in the soils. Now these phytohormones then develop what's called systemic resistance, and the bacteria and the fungi can no longer sort of take in those nutrients, because the plant, again, the mechanism is still a bit understudied here, but the plant can recognize that these are bad bacteria, bad fungi, and I'm not going to feed those. I'm only going to feed the ones that are helping me. So it's actually quite smart how the plants do this. But the exact sort of mechanisms and genetic releases are still unknown. And then you've got actinomycetes. So actinomycetes are halfway between bacteria and fungi. They're kind of like a middle ground. And in both structure and function, they kind of do a bit of both. Now, actinomycetes are known for what's called pathogenic antagonism. Now, they'll try any way they can to stop the growth, the reproduction, or even the function of pathogenic microbes. And this can be done by, again, all the previous listed ways. So systemic resistance, nutrient competition, parasitism, so they can actually eat the fungi or the bacteria themselves. But the most common way is antibiotic production. So they take in the nutrients, and then as a byproduct, they produce these antibiotic compounds, which then attack the pathogenic bacteria. That being said, it's not always specific to just the pathogenic bacteria. So sometimes this antibiotic production can affect the positive microbes as well. So then we're moving on to sort of soil organic matter and carbon storage. So microbes, their first and foremost role is decomposition. So you've got your bacteria decomposers, and they tend to be the first ones to have a crack at your soil organic matter. So the most common that I found in literature is Bacillus subtilis. It basically has an enormous array of functions, but one of its big time players in decomposition of soil organic matter. Fungal decomposers don't necessarily directly always try and decompose the organic matter to begin with. They like to feed off sort of root exudates. So it's different carbon forms that are secreted or exuded by the roots. And then they utilize this to produce enzymes. And these enzymes then act on the soil organic matter, and it further breaks it down. At the end of all this, you can still have some complex soil organic matter left in the soil. And this is where actinomycetes come in, and they can basically further decompose the organic matter, or if it's a complex carbon form, they'll act on that. This ends up with a soil organic carbon pool in the soil, and plants can take this up, and other microbes utilize this for their functions to go on and do other things. Now, there's some debate amongst sort of soil carbon experts about the manner in which this soil carbon exists. So you might have heard humic substances, and they're starting to debate whether those substances are actually a final resting place. What they think is it's more likely that the type of carbon that ends up in the soil is forever changing. It's fluid. It doesn't sort of finish in a final spot and goes, that's it, I'm just going to stay right here. As you can see, that the soil carbon is used by other bacteria, and there's a lot of different processes and different forms of carbon for it to end up as organic carbon just sitting in the soil. Some of that carbon is lost in the soil from leaching, so it doesn't always stay. Just because you started with this amount of carbon doesn't mean you're going to end up with that amount of carbon. All of these processes, microbes go through what's called microbial respiration. So they respire like us, and they produce carbon dioxide. So this then gets released into the atmosphere. Again, the whole, you've probably heard a lot about carbon sequestration and how good it is. If you suddenly increase the amount of carbon sitting in your soil rapidly, the microbes go, especially the R stratus, which I'll talk about a little bit later, go, thank you very much, I'll take all that carbon, and they rapidly sort of reproduce and expand, and then you get this influx of actually CO2 in the atmosphere. So that's because the process of them breaking down that carbon, they actually produce carbon dioxide as a byproduct. However, for the plants, this is really good because then they take in that carbon dioxide and they release it as root exudates, and we start the cycle all over again. So this is known as carbon turnover. So it's sort of the forever moving parts of carbon in soil. So in terms of nutrient cycling, carbon is obviously one of them, but another big player is nitrogen. So in your soil organic matter, after it's been decomposed by bacteria and fungi, you end up with organic nitrogen. And this is generally in the form of sort of genetic materials, nucleic acids. This is then mineralized by bacteria into ammonium. And however, other microbes utilize ammonium for their processes, and so therefore it's then immobilized back to organic nitrogen. Now, the reason why they use immobilization is because plants don't take up organic nitrogen, they take it up in the form of ammonium. So it's their most common usage of nitrogen for uptake. Ammonium can then be go through a process called nitrification, and this is done by a particular set of bacteria called Nitrosomonas. And then that you end up with nitrite. Now, nitrite, like some of the carbon in a carbon pool, can be lost through leaching. But further nitrification, so if it's gone through one set of nitrification, another bacteria called Nitrobacter then acts upon that and further nitrifies, and we end up with nitrate. This nitrate can then be assimilated into the roots. So although plants prefer to take it up as ammonium, they also do take up nitrogen through nitrate. At the same time, proteobacteria can then act on that nitrate, and it's a pretty complex process, but it's called dissimilatory nitrate reduction to ammonium. So this nitrate is then converted before it gets taken up by the plants back to ammonium. And then so it's just like an added steps for the plants to access this nitrogen. Then there's denitrification. So Pseudomonas denitrificans, they've really scraped the bottom of the barrel for the naming of that one there. Basically, we end up with atmospheric nitrogen. And this is not the end of the nitrogen cycle. So this atmospheric nitrogen can then be fixated directly into the roots by rhizobium, and that's probably your most common. So your legumes have these sort of bacterial nodules on them, the rhizobium nodules, and they fixate the nitrogen directly into the roots. But there's other ways as well. So there's a free living fixation, which is called Azotobacter. Azotobacter, rather than fixating it directly into the roots, converts the atmospheric nitrogen and turns it into this ammonium in the bulk soil, and then the roots take it up. So it's just an added step. And as you can see, that the microbes are doing everything they can in some way to get the nitrogen into a form that the plants can take it up, or turn it into atmospheric nitrogen, and then the plants will either directly fix that, or it'll end up back in the soil or back into some other organic matter, which will then go back into decomposition and back into sort of organic nitrogen. Now, soil structure is a big one. It wasn't until the last probably a couple of decades that they've realized that microbes are so involved in the structure of our soil. So a lot of it was, the credit was given to plant roots. And yes, plant roots do a large amount of stabilization of soil structure, but soil aggregation, a lot of that work is done by microbes. So plant roots exude different forms of carbon in root exudates, and obviously through the breakdown of soil organic matter, you've got different soil nutrients sitting in there. And these are acted upon by various microbes, both bacteria and fungi. And then those are broken down through the decomposition. They're broken down into secondary metabolites. And these are then used as binding agents or coagulants. And what you end up with is your soil aggregation or an aggregate. And these form sort of little pockets of clumped nutrients, but also water, basically anything a microbe needs to sort of live and survive. And same with the roots. The roots like to tap into these aggregates. Another way that soil structure is affected by microbes is through fungal enmeshment. So you've heard of hyphal networks, but fungi don't necessarily have to form these long linear lines. Sometimes they form like a mesh, like a gauze mat. And basically what they're trying to do is they're trying to increase the surface area to volume ratio of the plant root. So they'll tap into that plant root and they'll spread themselves out as thick and as wide as they can. And basically that allows, if there's a nutrient-rich pocket in the soil, they'll just completely surround that, and that plant will have constant access to that nutrient. And the fungi actually operate as fantastic gatekeepers, and they sort of take in the nutrient as much as the plant needs. They won't take any more than that. They don't take any more than they need either. So these enmeshments are fantastic for plants. However, as we were speaking earlier about tillage, these enmeshments can be just sliced in half through tines going through the soil. So sometimes it's good to break up the soil and give access to this carbon storage, but sometimes these hyphal networks have taken time to develop over time, and then we've just cut them in half. So, sometimes it's a win-lose situation, and it's up to the specific grower, and depending on what kind of soil they're dealing with, whether it benefits them to turn that soil over or not. The last sort of key function for microbes is soil water availability. Now, that aggregation that we're talking about obviously allows these pockets of air in the soil, but it also increases the ability for water to move through the soil. So if you have a hard top of soil that hasn't been affected by microbes, that water's not going anywhere. Sometimes the plant roots can't sort of access it. Whereas if you have a nice porous soil, water moves fairly freely through the soil. Now, it's not only plants that need water to function. Microbes are more often than not hydrophilic, so they like water. You do have a few hydrophobic microbes, but again, they're more extremophiles, so they're not necessary unless the environment sort of promotes their growth. And there's a varying amount of sort of water availability that microbes benefit from. So again, like your carbon inputs, your nitrogen inputs, all that sort of stuff. If you have a climate that is, high rainfall, then the microbes that are more hydrophilic, so the ones that love the water more often, are going to thrive. If you have a more sort of a drier region or climate, then the ones that don't like a lot of water and are more resilient to utilization of less water, then they're the microbes that you're going to benefit. And this goes back to me saying about how people will just put any microbe in any soil and hope for the best. You actually need to specifically target what your climatic conditions are for the soil, because your microbes may not survive what you're adding in. So I want to talk about fungi and bacteria, mainly because they're the two major players in the soil, but there are other microbes at play. And so your protozoa are very important they feed on bacteria and fungi. So they're sort of not just the pathogenic bacteria, but they do feed on sort of the beneficial ones as well. But it is important for protozoa to exist so that bacterial predation, the feeding on the pathogenic bacteria and fungi to stop their growth. And then by feeding on these, they allow for a different form of nutrient to be available to other microbes. So they eat the bacteria and then convert the cells into sort of bioavailable nutrients. Algae are very important. They're seen as the pioneers of the soil world. Algae are autotrophic. They are themselves a source of carbon and nitrogen for other microbes. So they sort of attach themselves to anything they can, and the algae will grow, and then the bigger microbes will then feed on the algae. And then nematode, again, nematodes are very similar to protozoa in the way they act. They're very important for sort of the regulation of bacteria and fungal populations in the soil. But also, because of their size, they do affect soil aggregation. So they create these pockets by sort of moving around in the soil and pushing the soil around so that they can expand themselves. So spatial influence of soil microbial diversity. So again, depending on where you put your crops in and how big a root system your crop has, and also how close your crops are together, will influence the microbial diversity that you have. Within just a single plant in the soil, you have the bulk soil, which is around the outside, and that will have, again, one certain type of microbes that only function with nutrients in the bulk soil. Closer to the root is the rhizosphere. Now, this has been known. So Lorenzo Hiltner was talking about the rhizosphere in 1904. So the idea that microbes have been involved in plant growth is over 100 years old. Now, new light has come to what's called the rhizoplane. So this is basically the immediate sort of soil particles or nutrient particles that are semi-attached to the roots but aren't necessarily penetrating the roots. They're basically the sort of like the gatekeepers of nutrient release to and from the root itself and the rhizosphere. And last, the endosphere. So the endosphere are more like your arbuscular mycorrhizal fungi that sort of attach themselves not only just around the root but into the root itself. So they actually penetrate the cells of the root to achieve sort of nutrient exchange. temporal influence of soil microbial diversity. So again, this comes back to soil microbiologists trying to marry up terms with sort of terrestrial ecologists. So there were, before sort of looking at it as a time point of view, it basically was copiatrophs, which is microbes that benefit from large amounts of nutrients, to microbes that can function with like low amounts of nutrient availability. So previous to looking at it as like an R strategist or a K strategist, it just used to be about nutrient availability. Nowadays, they're trying to utilize the same terminology as you would with terrestrial animals or plant life. So our R strategists are the ones that grow very quickly, very early on. They need lots of nutrients, but they're not very good at sort of fighting for those nutrients. They tend to be in large numbers, and they don't require a lot of energy to reproduce, to do their function, because they will just overpopulate themselves. So their expenditure is low. K strategists, again, are your longer developing microbes. They take a while. They're very good at competing because they will utilize the smallest amounts of nutrients to reproduce and to do their function. There's not as many of them because they're so efficient at their job. But however, they do tend to take up more nutrient in terms of for their sustained life. So because they live for longer amounts of time, they tend to be more expensive in terms of, to keep them going, you need to put in more nutrients. So new technologies have allowed us to sort of shift into different ideals of how we approach microbial research. So we have culture dependent versus independent. Now, only up until the late 90s, people were still taking soil samples, taking a bit of and streak plating, growing the microbe out on an agar plate, and then testing that microbe. Nowadays, we can literally just take a soil sample, we can look at the nucleic acids and go, this is the microbe that is in that soil. So that's a huge jump in the way that we look at what microbes are there. This genome sequencing technology is forever improving and becoming more accurate. The computational power, however, to match that and to figure out and do the calculations of what's there and what's not, and to say with certainty whether there's ever any overlap in the genetic material, is lacking. And so we're at a stage now where we have sort of gene sequencing technology that needs a supercomputer to be able to run it and say with absolute certainty that that's what's there and that's what isn't. There's obviously human error involved as well in these samplings, but the biggest limit to in terms of microbial diversity is the computing power to run the statistical analysis of those samples. Before we were talking about sort of the function of soils. So there's bioassays now that exists where you can literally take a section of soil and quickly run it on a microplate, or there's a box called MicroResp, and that basically will tell you if the microbes in that soil are respiring. So if they're doing their job, if they're functioning, as I showed you earlier, that they're producing that CO2. MicroResp can calculate how much CO2 is being produced. So if you know how many microbes are there, or how much or what type are there, and you measure that CO2 production, you can actually figure out how efficient those microbes are at doing their function. It's hard to tell sometimes what exact function that is, but at least you know that they are alive, they are doing some kind of job, and by how much you can tell by the amount of CO2 that's produced. This is just a little bit of a snapshot to give you an idea, and I don't want to scare you too much, but this is like a needle in a stack of needles. So this is a meta-analysis paper that I had a look at, and these guys just looked at one lot of bacteria, three different phylum, which is like pretty high up in terms of the bacterial domain, but they only looked at three. And then within that three, they only looked at a few classes. And then within those classes, these are the amount of species that were involved in different crop types. So as you can see here, your Bacillus subtilis is found in 3 different major crop types. And this is all Northern Hemisphere, but a lot of the times the bacterial species here in the Southern Hemisphere, if we're growing those same species types, we're going to tend to find the same bacteria on those plants. And as you can see, Bacillus is found in almost every type of major crop that can be grown. But at the same time, you can see variability amongst the type of bacteria that's grown. So sometimes specific bacteria will favor one crop over another. But in that same breath, you can get one crop type that will have, a huge array of different bacteria, all doing either the same function or a completely different function. So that brings me on to my next slide, is that the way we used to approach microbial research, and growers used to have this same mindset, was if you were to increase a population of one species of microbe, that you increase its function and then you increase the soil function. So the microbial function might be a specific step in the nutrient cycling process. The overall soil function is nutrient cycling of itself, or plant uptake of that particular nutrient. Now, if you increase one microbe population, there's no way of telling that it's directly involved. Like if you're unsure what microbe you're adding, or if the system will allow that microbe to do that. So sometimes both researchers and growers have gone, I just sprayed my crop with this microbial inoculant. Now look how much nitrogen I've got in my soil. You can't always say that specific microbe was doing that exact role. That the increase in nitrogen can be attributed to that one microbe. So it's actually a lot more like this. If you increase your diversity, if you increase the different types of microbes that you have right across the board, then you're going to increase the microbial functions. Now, as you can see here, multiple populations or species of microbes can be involved in one function. That same function one species can be involved in 2 separate functions. And then the overall soil function, which might be, again, going back to those key functions, so you get your nutrient cycling, your carbon storage, your biochemical regulation, that can be affected by a whole suite of different microbes all interconnecting. The last part is what we're starting to realize now is that microbes can actually pass on genetic information sideways. So they can pass it on from one species to another without having to reproduce. And this is huge in the microbial space, because if one microbe has learned to do something and another microbe is also involved in that same process but are lagging behind, but they aren't too dissimilar in terms of their genetic makeup, but they are different species, that microbe can pass on that genetic information to a different species and teach it how to function better, or how to better adapt. Or it may be a regulatory process and saying, oh, hang on, we've done our job, there's no more nutrients left, and it will pass that genetic information and going, we need to stop, we can't grow anymore because there's too much. So that changes the way that we look at what microbes that we need to put in our soil, because it's a much more complex picture than first thought. And so what the new approach to soil microbes is increasing its diversity will increase its ability to function in the soil and become more resilient to outside or external pressures. Just a quick overview of my experiment that I'm looking at is I'm going to inoculate some soils, and then I'm going to reintroduce some microbial communities to those to the sterilized soils. These sterilized soils will have no microbial life left. They won't be doing any activity because of the gamma radiation. Once I re-inoculate them, I'm going to see whether, and again, I'll have different soil types, and I want to see whether the soil type affects the microbial community over sort of 0, 7, 28, and 90 days. So at 28 days, I'm going to send off my soil samples to AGRF to see whether they're going to send me back the genetic information. And I'm going to see whether that microbial diversity profile has changed based on the soil that it was inoculated into. But all along that time period, I'm also going to be testing the functions of those soils and the physicochemical properties of the soil to see whether not only the soil type affects the microbial growth, but whether the microbes can actually drive and change their environment they live in. Because as you've seen before in the slides, quite a number of soil nutrients, the physical structure of the soil, water availability are all driven by microbes. So it's suggested that there's a feedback loop involved with microbes and the soil, and they eventually, given enough time, will reach some kind of equilibrium. I'd just like to thank sort of Soil CRC, Southern Cross Plant Science, the Centre for Organics Research, and the DPI for allowing me to do this research. Thanks, guys.
Thank you for watching. If you found this information useful, there are many more related projects to discover at our website. Please like this video and subscribe to our YouTube channel for more Soil CRC webinars, project updates, and conference presentations. This webinar series was produced by the Soil CRC, jointly funded through the Australian Government's National Landcare program, to build the technical capacity of natural resource management agencies, landcare and grower groups to deliver soil health information to farmers for improved soil management. For more information about our projects, visit soilcrc.com.au.